Cystic fibrosis is a type of hereditary disease, and when the both copies of the gene is mutated, it leads to cystic fibrosis. Cystic fibrosis is a disease in which obstruction of gastrointestinal tract and respiratory tract occur, and due to respiratory insufficiency, it results in the death of the individual at the age of 30. Now, to understand the cystic fibrosis, first of all, we have to understand what is CFTR. See, what is the full form of the CFTR? CFTR is cystic fibrosis transmembrane conductance regulator. It is a type of Cl- channel. It helps in the movement of the Cl- across the plasma membrane. Now, so CFTR contains two segments. Each segment contains 6 helices and 2 NBDs, nucleotide binding domain and a regulatory domain. Now, the regulatory domain contains serine. So, when the serine is phosphorylated by CMP dependent kinase, it allows the binding of ATP to the NBDs domain, that is NBD1 and NBD2. But when the regulatory domain, that is serine residue on the regulatory domain, is not phosphorylated, it does not allow the ATP to bind with the NBDs domain of the CFTR. Now, that is so when the serine residue on the regulatory domain is phosphorylated, it allows the binding of ATP to the NBDs domain, and it leads to opening of the channel. That is why Cl- moves across the channel. But when the serine residue is not phosphorylated, the ATP does not bind with the NBDs, and Cl- does not able to cross the membrane. So it is a closing state. In case of dephosphorylated of regulatory domain, ATP does not bind with the NBDs domain, and it is a closing state of the channel. Now, that in case of cystic fibrosis, 70% of the cases, the phenylalanine at 508 in the NBD1 domain is deleted. And due to the deletions of the phenylalanine 508, it leads to a different conformation of the channel, and this conformation does not allow the Cl- movement, and so it is a closing step. It remains in a closing step. It does not allow Cl- the movement of the Cl-, and that is why the water does not, the movement of the water is also diminished. In case of normal circumstances, the inner surface of the lungs contain mucus, and this mucus becomes thin and hydrated. So the cilia which is present in the inner surface of the lungs can easily sweep away the bacteria, that is staphyloccus aureus and the pseudomonas aeruginosa. So it is easy to remove those types of bacteria by the weighting of the cilia. But when in case of cystic fibrosis patients, the mucus becomes thick and dehydrated because the CFTR channel which is present in the surface of the lungs does not work. So that is why the mucus becomes thick, sticky and dehydrated. This thick, sticky and dehydrated stimulate the growth of the bacteria, or accelerate the growth of the bacteria, that is staphyloccus aureus and pseudomonas aeruginosa. And in this case, the cilia does not sweep away the bacteria from the inner surface of the lungs, and ultimately the damage of the lung occur, and due to respiratory insufficiency, the patient will die. So this is the cause of the CFTR. There are several other causes, but the main causes of the CFTR is the deletions of the phenylalanine 508 at NBD1 domain in the CFTR, that is cystic fibrosis conductance regulator. Thank you. So cystic fibrosis transmembrane conductance regulator is a type of Cl- channel. So cystic fibrosis transmembrane conductance regulator is a type of Cl- channel. It is also called as a CFTR. Now this is the plasma membrane two segment. It contains two segment, and each segment contains six helices. Each segment contains six helices. This is a NBD1 domain, and another is NBD2 domain. NBD1 means nucleotide binding domain 1 and nucleotide binding domain 2, and the green is the regulatory domain. Regulatory domain or art domain. Regulatory domain is contains serine residue. Now it is the inactive state. In this case, there is no movement of the Cl minus. But when the R domain, the serine residue on the R domain is phosphorylated, it allows the binding of the ATP. Now ATP binds with the NBD1 and NBD domain, and it opens the channels here the movement of the Cl minus occur. So as you can see, R domain is phosphorylated. Now this R domain, phosphorylation of the R domain allows the binding of ATP. As you can see, the NBD1 and NBD2 domain binds with ATP, and it leads to opening of the channel. Now this is R domain is also phosphorylated. Now movement of the Cl minus occur because it is a opening state. But when the ATP is hydrolyzed into ATP and PI, and the phosphate from the regulatory domain is removed, channel becomes inactive, or it is remain in a closing step. But in case of lungs, the inner surface of the lungs contain mucus. In normal circumstances, it is a thin, and the cilia which is present, it easily sweep away the bacteria. But in case of cystic fibrosis, the mucus become thickening. So thickening of the mucus due to diminished the movement of the water into the inner surface of the lungs. That is why thickening of the mucus occur, and it provides the growth of the several types of bacteria, and a cilia does not sweep away the bacteria which is present in the surface of the lungs, and ultimately